Hello, everyone, and welcome to your new chapter. Well, same basically theme, but different chapter, which is more autoimmunity. So, more basically the factors that can cause autoimmunity. And also, we're going to be talking about transplantation, which I think they go really hand in hand. So, in the previous chapter, we spoke a lot about, in per se, but peripheral and central tolerances, these kind of tolerances that we have to teach our T cells and B cells not to be self-reactive, which is extremely important. And we learned that what happens when one of these processes are being basically disrupted. We get autoimmune diseases, and then we went into the pathology of how these autoimmune diseases can actually occur. So autoimmune diseases are very complex. It's not that you have just one factor that can cause it. Like it's not that just you have one genetic factor that can cause certain autoimmune diseases. There's in some cases that there is, and I'll be talking about them. But in the majority of the cases, it's not just one single factor. It's a combination of genetic factors, but also infection and or environmental exposures that can cause basically this immune regulation and specifically affecting our peripheral tolerances or affecting basically our central tolerance that can induce an autoimmune disease. So now let's look at basically the genetic components. This genetic component is a factor that can induce actually autoimmune diseases. So we see this a lot in generally with basically families. You start to see that in certain families there's a autoimmune disease that's being always popping up. That is always indicative of a genetic factor. But we see this mostly a lot in per se in your strains of mice. So within mice, you have different strains. Every single strain has a different genetic background, but within one strain, they have an identical genetic background. So, for example, you have the black mice that we call them C56 black. They all have literally a identical genetical background because they're inbred. Their genes are kept the same. And basically you have this other strain that's called nod. These nod strains, these are again all strains of a mouse that are genetically identical. So what we see with these ones is that these mice have a predisposition for type 1 diabetes. For some reason, these mice, almost all of them develop type 1 diabetes the more they grow, which is crazy. So we see here basically these kind of nod mice. This is the incidence of the diabetes in these nod mice. And you see that basically by week 13, you start to see an increase of type 1 diabetes. So what we see is that in female, which is shown here in red, it increases much more than in male, which is shown in blue. So you, other than just seeing, first of all, these mice getting type 1 diabetes, you start to see that it's much more sex oriented in a sense that females are much more predisposed to type 1 diabetes in these mice. And in general, in humans, it's the same thing. Usually, autoimmune diseases is actually, you see it much more in women than in men. This also is another factor to, or kind of, let's say, kind of proof to show that there's a genetic component to autoimmune diseases because this is linked to your X and Y genes. So you see this much more happening in females, the autoimmune diseases, than basically than males. So we see here that by the age of 25, basically almost weeks, you actually have almost 80% of your female mice having type 1 diabetes. So you see that there's a genetic component that almost all these mice are getting it, and they have exactly the same, basically, genetic material. Like, it's definitely just something genetically. But what's something that we see also here in this kind of graph that is weird is that why is this one mouse over here having it at basically 13 weeks, while the other one is having it at 25 weeks? Why? They have the same genetic components. Why is one getting it at day basically week 13, the other one's at week 15? It's most probably the environment. Something in the environment is causing that this mouse is getting at week 13 while the other one is at week 25. So this is where I'm telling you that it's not just sometimes one factor. The majority of the times it's a genetic factor. You have some predispositionally for autoimmune diseases, and then you're basically the environment or an infection can push it even more. So it's usually a combination of these. And this is what you see here. This is a beautiful graph to show you both that there's a genetic linkage, both that the strain of mice has an increase of type 1 diabetes, two, that the basically is much more predisposed in females, so it's much more in the X chromosomes, and technically also that there's an environmental factor because that's why not all of them are spontaneously getting type 1 diabetes at the same time. And we've done a lot of these basically genetic studies to basically autoimmune diseases. And of course, our understanding of it, of this kind of genetic factor, has exponentially risen, all thanks towards basically the ability to sequence genomes. Because we're able to sequence genomes, we started to see actually risk factors. So here, this beautiful graph, we have basically in these big circle colors, you have basically different autoimmune diseases. Like you have psoriasis, and basically this is bowel disease, inflammatory bowel disease, type 1 diabetes, rheumatoid arthritis, multiple sclerosis, systemic lupus erythematosus, different autoimmune diseases over here. And we see these basically are connected to these other bubbles, and these small bubbles are genes that have shown that these can give a predisposition towards whatever they're connected to. So, for example, HLA-C gives predisposes PSO, predisposes a human being to basically psoriasis. Or you have here basically, and here, let's say interleukin 1 receptor 2 gives you basically inflammatory bowel disease. What you see also that's also cool is that certain genes are predisposing you to one, let's say, autoimmune disease. Well, you have certain genes that predisposes you to multiple. So, for example, HLA-DRB1 predisposes you to SLE, to MS, to RA, you name it. So you do also see that certain genes can predispose you to one type, but some can also predispose you to multiple types. So we started to see multiple type of genes that are being affected, and these are basically the ones that I think you should definitely know. So I definitely want you guys to know these ones. So we have these genes like IDE. Remember IDE, guys? IDE is basically this gene complex, or this protein complex that basically is found most in your mesenchymal, basically thymic epithelial cells. And this IDE, what it does, it increases your tissue restricted antigens to be presented, and this drives your negative selection in your thymus for T cells. So this is basically responsible for making sure that no self-reactive T cells are being actually released into the periphery or your blood. So if you knock out IDE, you remove all this negative selection because you don't have any more of these tissue restricted antigens. So negative selection is gone. So you have a lot of self-reactive T cells going everywhere, and this causes basically in the human disease it's called APS syndrome, and this you have multiple different types of um, autoimmune diseases, multiple. So this is basically where you see one certain genes causing multiple types. CTLA4. CTLA4 is a co-stimulation molecule that induces anergy, and these are found in your Tregs. So Tregs use CTLA4 to push T cells into anergy. It kind of stops basically dendritic cells to present any antigens. It stops everything. So if you remove CTLA4, you're removing a lot of the peripheral tolerance. So you actually have Graves' disease, type 1 diabetes, and other types. So it's multiple. FOXP3. FOXP3 is the transcription factor found in Tregs. You remove it. You remove your Tregs. You remove almost basically all of your peripheral tolerance. FAS. FAS receptor, these are the death receptors. So we learned that basically in the periphery, if a reactive T cell or B cell actually is able to react with a self antigen very strongly, it basically goes into death. And how does it do that? Through the upregulation of FAS receptors. If you remove FAS receptors, they are not able to remove self-reactive B and T cells. So you have basically this kind of ALPS syndrome, which is again basically very similar to this. You actually have multiple types of immune diseases. C1q. C1q is basically a protein that is found in your classical activation of the complement cascade. So the complement proteins, they use C1q to activate your classical pathway. So if you remove C1q, you're not able to remove certain basically immune complexes or apoptotic cells. And these apoptotic cells or immune complexes can be many of them can be self-antigens. If you don't remove them, you're increasing the probability of self-reactive T or B cells to touch them and get activated. So this is how C1q. And this can cause systemic lupus erythematosus. So you see here one basically gene causing this kind of disease over here. And we also see overexpression and underexpression of different cytokines. Certain cytokines can induce or proliferate a lot of self-reactive B or T cells, for example, like TNF alpha, for example, or interleukin 3. Or it could be that sometimes that despite, for example, TNF alpha, if you overexpress it, you get IBD, but if you underexpress it, you get lupus. So sometimes it's not so very black and white kind of scenarios. Sometimes you need a perfect amount of it. That in a sense that if you push too much TNF alpha, you have a lot of self-reactive T cells. If you underexpress TNF alpha a lot, you do not have the clearance of certain basically apoptotic cells or other immune basically kind of, let's say, complexes that have self-antigens. So sometimes you need a perfect balance between that. So I don't need you to remember these at all, but I definitely want you guys to know these for sure. So in the GWAS studies, so what a GWAS study is, it's basically what we call a GWAS. It's a large-scale genome-wide association study. So what you're doing is you're taking literally multiple genomes of many human beings and sometimes even population scale, and you're looking to correlate the frequency of a disease with genetic variants. So these genetic variances are usually typically single nucleotide polymorphisms, SNPs, we call them, which is technically you're changing an amino acid. So it's literally single amino acid mutations, or single nucleotide polymorphism. So basically, it's a change of a single nucleotide. Sorry, that's more than what a SNP is. It's literally one single nucleotide polymorphism. So you're changing one nucleotide that could actually cause a change of an amino acid, but sometimes not. Sometimes not really. But this SNPs, basically, what you're trying to look is for genetic variants, these kind of nucleotide polymorphisms that occur, and you're trying to correlate if you see a polymorphism, does that actually increase the frequency of a disease? So here you have a beautiful GWAS correlation with Crohn's disease. This is basically a inflammatory bowel disease that you have. Basically, you see these SNPs, for example, interleukin 23. Here you have a SNP in this gene, in this other genes, and you see that you actually have so many different types of SNPs that can increase Crohn's disease. Well, we did that basically for many different types of autoimmune diseases, like including Crohn's disease, and we see a lot of basically kind of similarities. For example, we see CTLA4, which is found mostly over here, this ATG16L1. This is the gene that encodes for the protein CTLA4. You see it in Crohn's disease that, for example, if you have an increase or a polymorphism there, it increases the likelihood of getting Crohn's disease. But not just Crohn's, you also have it in type 1, Graves' disease, Hashimoto's, rheumatoid arthritis, MS, you name it. Because you're really removing Treg function. Because Treg, they really use CTLA4 to push T cells into anergy. So this is how they do this really, really well. So this is how actually GWAS analysis has helped us to identify certain basically genes or basic pathways related to autoimmune disease. But we also see, for example, if you go back here, you see interleukin 23, there's basically interleukin 12, there's CARD9, MUC9, NOT2. If you look at them, these CARD9s and PETGR4s, these are basically all found in your, basically the dendritic cells. And these are activated when your dendritic cell is able to identify a PRR, a pathogen recognition, a PAMP, a pathogen associated molecular pattern like beta glucan, and that binds to your PRR, this pathogen recognition receptor, lectin-1. This activates your CARD9 and this PETGR4, which causes a massive upregulation of interleukin 12 and 23. So you see that 12 and 23 are pushing T helper 1 and T helper 17 cells. So you see how, for example, CARD9, PETGR4, interleukin 12, 23, how having SNPs those by causing them to be, let's say, increased by much more or be overactivated, you're pushing T cell overactivation. And we see this a lot, where a lot of SNPs that are pushing literally T cell overactivation, like T helper 17 or T helper 1, we get there actually a predisposition to autoimmune diseases. So we see from this GWAS, we can put these genes into many categories. In some categories from coming into the availability of autoantigens and clearance, for example, the C1q or IRE. C1q really removes the clearance of autoantigens, but also we see that IRE also kind of provides the availability of autoantigens in a sense that you need to use autoantigens to actually drive negative selection. Without it, you don't have it. We see in apoptosis, like the FAST receptors that I spoke about. Without the FAST receptors, self-reactive B or T cells cannot be removed or dead or basically induce apoptosis. Signaling thresholds, certain signaling kind of induce the overactivation of T cells. Expression So cytokine expression or signaling. So again, this is all falling in the T cell overactivation or B cell overactivation as well. Costimulatory molecules, we see that in the CTLA4, if you remove it, you really are pushing Tregs not to do any more anergy. So you're not giving this inhibitory co-stimulation, but you also see in co-stimulatory molecules like B7 or CD28, if you see an upregulation of those, you definitely can induce yourself into autoimmunity. And of course, Tregs, regular T cells, if anything happens to them, this can really have detrimental effects when it comes to autoimmunity. So, a lot of autoimmune diseases are controlled by not just one gene. It's sometimes a combination of multiple genes. So, for example, you see here with Crohn's disease, there's multiple genes that can cause it. It's not just this one gene that can cause it. However, there's sometimes a case where one gene can cause literally a autoimmune disease, and we call these basically these monogenic autoimmune diseases. Hence, where one gene can predispose you to autoimmune diseases, and these are the ones that we really spoke about, like IRE. If you remove IRE, you're removing negative selection. CTLA4, you're making Tregs not being able to induce anergy. FOXP3, you're removing completely Tregs fast. You're removing the death receptors that are inducing apoptosis of these self-reactive B and T cells. C1q, so you're not helping your complement system to remove these basically autoantigens. So this is where we see monogenic defects. But one thing that we also see that's super interesting is MHC, because as I said, antigens are being presented to T cells through MHC. So sometimes what we see is that MHCs are also carrying the self antigens and presenting it to T cells. And the thing with MHC is extremely, how can I put it, very polymorphic in a sense that we don't have a human. It's very hard to see human beings that have exactly the same MHC. So, as we know, we have two MHC classes, MHC2 and 1. 2 is more for the antigen presenting, so this is much more for basically the outside antigens. MHC1 is more for the inside antigens. So we see that MHC1, which is made of the alpha chain and the beta-2 microglobulin, it's made of 3 genes. So to get actually an MHC1, you need 3 genes. You need HLA-A, HLA-C, and HLA-B. These 3 genes make up an MHC1. Well, there's so many different types of alleles, meaning different types of genes within the HLA. With HLA, you have 8,000 different types. B, you have 9,000 different types. And basically, C, you have 6,000 different types. So when you're born, you get one HLA type from your parents, one type from basically one B from your parents, and one C. So you have, for example, HLA-A 1,200, HLA-B 22, and HLA-C 3,664. So this is then your MHC1, which then gives you different combinations, and hence why different people have different HLAs. This is what we call HLA as this major histocompatibility gene, because there is a compatibility, because we have different MHCs. So we're not compatible in a sense. We have different combinations. And same thing goes with MHC2. MHC2, we know, is made of the alpha and beta chains that are coming from genes coming from the DP, DQ, and DR. So these are the ones, and we have different multiple types of them, basically. So what we see that's super interesting, I have to say, is that certain people with certain basically HLA genes, for example, in this case, the B27. So this is basically what it means is that in this case over here, you have the HLA-B number 27. If you have that allele, you have an increased risk of ankylosing spondylitis, because you have an MHC. So this specific basically gene that's giving you a product of an MHC that is able to bind much better or to a higher affinity to self antigens, causing ankylosing spondylitis. Same thing goes to type 1 diabetes. If you have the DQ2 or DQ8, these basically can give you an increased affinity towards actually binding to self-antigen much more. So different MHCs, different combinations of MHCs gives you different affinities. And certain MHC genes gives you higher affinities. These are examples. But to make it a bit more complicated, what we see, of course, for example, we see that people that have the DR and DR4 alleles, so these are the MHC2, we see that they have a predisposition, but there's something called polymorphisms, meaning that people that let's imagine that you have, let's say, 20 people that have the DR3. They all have the same gene, but they have basically SNPs. So some of them have polymorphisms, like one or two amino acids that are different within the same DR3 gene. These are called polymorphisms, and this can cause even much more predisposition. So imagine you already have the DR3 gene. This DR3 gene already has a high affinity towards a type 1 diabetes, towards, let's say, your pancreas. So it's able to bind to self antigens coming from your pancreas at a much more higher affinity than, for example, DR5 or DR2, for example. What they see with the DR basically 3 is that sometimes they have certain amino acids that are being basically affecting, are being basically there's a SNP that usually occurs in the beta alleles, so mostly in the beta chain. So this DR3 is basically also presenting is encoding for both the alpha and the beta chain, so it's mostly in the beta chain. So you see this in position number 57. You see that basically in general it's an aspartic acid that creates this beautiful, literally kind of salt bridge. It creates this beautiful basically salt bridge with the basically. So this is basically the aspartic acid over here. This aspartic acid, it creates a beautiful salt bridge with the alpha chain over here. So the blue is your beta chain, the basically gray is your alpha chain. You see that this aspartic acid is causing this nice salt bridge. Sometimes in some people that have the same DR3, they have basically a SNP that removes now this aspartic acid into basically an alanine, and this alanine doesn't do anymore that salt bridge. So what happens is that now this MHC basically that is already has a high affinity has even a greater affinity. So you can go from genes level, so different basically alleles, to SNPs, to literally one amino acid within that allele that can cause even a much more higher increase of binding to self peptides. So this is how much MHC also is a huge contributor in the genetic factors, because they're the ones presenting your antigens. If you have an MHC that is really pushing or binding so well and presenting a lot of these self peptides, well, you're going to be predisposed much more. So this could coming from the type of HLA allele, or even the basically polymorphism, so the SNP in within that MH basically allele that you have. So now let's go into more the environmental, which I feel is much more interesting and cool. Environmental factors, one of them is basically actually how you're born. For some reason, we've started to see that babies actually delivered through the C-section are much more predisposed towards autoimmune diseases compared to babies given through the vaginal delivery system, basically. And this is mostly because of the microbiota. So the microbiota is the bacteria inside your gut. So that has a huge influence on how your immune cells are reprogrammed. And I'll be talking about this a bit more right now here. So gut and autoimmunity. So as I said, within your gut, you have much more bacteria in your gut than you have cells by one log more. That's a huge increase. Just imagine how much of an increase that is. So within that gut, you have many different types of bacteria. It's literally so many types. And you can basically put them in two categories. Either you put them as beneficial bacteria, ones that really help you live much in a safer manner or more healthier, and ones that are much more, let's say, kind of damaging, more pro-inflammatory. Sometimes what we see in the case, basically, in this kind of scenario, you basically are colonized with bacteria that are much more pro-inflammatory. Because they're much more pro-inflammatory, they're literally making your gut lining much more damaged. You're damaging it, and you create something called the leaky gut. What that leaky gut literally means is that you're causing your gut to actually open up, and so much bacteria from your microbiota are releasing into your blood, and this really can cause so many issues. This release of your basically bacteria in your gut to the outside, because now your gut is much more open, it's more leaky, it's allowing them to go out. It can do three things. A, sometimes these basically bacteria have certain proteins that resemble host proteins. So they resemble self-antigens. And they can cause a confusion to your T cells, because now your T cells are activated, they're trying to kill your, let's say, your leaky microbe, but it looks like your pancreas. I was like, okay, I'll kill the bacteria and then I'll go kill the pancreas because it looks like it so could be an invader. So this is called molecular mimicry. So it mistakenly your T cell that now is basically killing basically an antigen that is found in your basically bacteria that's invading you looks now also very similar to a peptide that's also found in your pancreas. So this is what sometimes happens. What also happens is that and when you have so much pro-inflammatory basically bacteria, you're increasing the T helper 17 cells much more, and you're reducing Tregs. So you're pushing much more the T over activation. And basically, you're also what you have is that in these beneficial bacteria, they release this we call them the short chain fatty acids, and these short basically so basically, these short, yes, chain fatty acids, what they do is they really are very good at stopping out, basically, immune responses. They really stop, and they are anti-inflammatory, basically, molecules. So what happens is that they basically have, if you have a lot of harmful pathogens, you reduce the amount of this basically short chain of fatty acids. So this is how, for example, this is related to each other together, the gut and basically the way you're basically delivered. Because if you deliver to the C-section, you have a different type of bacteria. Mostly of them are more pro-inflammatory, while the here you have a much more, let's say, you have more healthier bacteria that are not that inflammatory. Another one that is so interesting is the hygiene hypothesis, which basically means this I'm quite sure you've heard. Imagine that you have basically twins, okay? So genetically they're exactly the same. If you put one twin basically in the farm where that twin is constantly predisposed to so many microbes and dirt, and then you put someone in the city in a very clean apartment, you see that that basically the twin in the clean apartment has much more predisposition to autoimmune diseases, way much more, and we see this a lot. Epidemiological studies have shown that, and where basically if you're in a place that is in areas that are industrialized and much more cleaner regions, you have much more chances of getting autoimmune diseases like type 1, Crohn's disease, and MS. So, what you see is that if you are more dirtier, so for example, if you're more exposed to microbes, for example, on farms, you have much more incidence to these diseases. This is the hygiene hypothesis. And a big reason we think about this is because when you're exposed to so many these kind of, let's say, disease, you're much more exposed to, let's say, microbes at a young age. Your your immune system gets taught much more, and it's able to kind of not overreact because it's being taught how to deal with it at a young age, and you start to develop a lot of more Tregs. If you're not, let's imagine as a baby you're extremely clean, and then you're basically way later on exposed to basically other microbes that you're not used to, your immune system can overreact because they're not used to it. They have not seen this before, and this can actually cause autoimmunity. Another one is vitamin D, and they've seen this a lot. That actually, people coming from the north of Europe, like for example, coming me from freaking Finland, there's a much more predisposition for autoimmune diseases compared to people coming from countries from the Mediterranean, like Lebanon, like Spain, Greece, Portugal, Italy, you name it. These guys that actually get a lot of vitamin D from the sun, they do not have so many types of autoimmune diseases like MS or type 1 diabetes. Because what we see with vitamin D has a very beautiful action in our immune system. It's able to actually stop basically the dendritic cells presenting self-antigens to CD4 T cells. It can stop self-reactive B cells. It can also help in basically stopping T helper 17 cells as well. So it really helps a lot in basically causing more, let's say, stability and more this kind of Treg functions, stopping T helper 17 cells, stopping autoimmunity, because it can actually stop sometimes a over exaggerated autoimmune, basically an exaggerated immune response. And another one is infection. So there's two ways how an infection can cause autoimmunity. One is that basically once you actually have a infection, it can actually cause a huge increase of co-stimulation. Basically, if you're infected with a virus or a bacteria, you actually have an increase of technically B7 and CD28. So there's much more co-stimulation, and this can cause T cells, self-reactive T cells or B cells, to start literally survive and to actually start proliferating. Two is that what you see when you're bacteria viruses, you have a lot of damage to yourself, to your basically own cells. The more damage you have to your cells, the more release of self antigens, and this is really bad. And three, you disrupt your immune suppression. Basically, when you're putting you have a virus or bacteria, you're really causing your T cells to really go haywire. You're stopping your Tregs because you don't want them now. You want the T helper 17. You want the T helper 1 cells. You want your cytotoxic T cells. These really reactive T cells to be very, what do you call it, active. And sometimes some of them could be self-reactive. And another one that is super cool that we spoke about is molecular mimicry. And I'll give you an example of this molecular mimicry. You see this a lot with Streptococcus. So let's imagine you have basically you're infected with Streptococcus. You basically have your B cell response, which creates plasma cells that are producing basically these antibodies. You have two different types of antibodies being produced. Antibodies binding to this red triangle over here, found in your Streptococcus, and other antibodies found basically in your this kind of green little oval shaped protein. What happens sometimes is that these antibodies, when they pass through your heart, they will actually bind specifically to an epitope that is very identical. So these literally triangle orange that are they're not the same, but they're similar in structure. And this antibody that is supposedly bounding to the Streptococcus is able to bind to here because it's so similar. This is what we call molecular mimicry. This blue one that doesn't bind anywhere because there's nothing that mimics basically this protein is basically filtered. This one that is mimicking this molecular mimicry, the molecularly wise, they're very similar to each other, to this red triangle and this orange triangle, that you actually have this antibodies now binding to your heart. So you have this kind of rheumatic fever, which is basically an inflammation in your heart. That's extremely dangerous. This is how Streptococcus, for example, can cause autoimmunity for us. Another thing that is super interesting, this will not come in the exam. This is just a nice case study for you guys to know. They saw this a lot, specifically in 2009, we had this kind of pandemic that was basically this H1N1. Basically, sorry, not pandemic, epidemic. That was basically this winter influenza. Sorry, actually, it was a pandemic. H1N1 winter influenza pandemic that occurred in 2009 in China. And they started to see that there's basically this kind of narcolepsy type 1, which I'll talk about what that is, this disease that suddenly was like, you have like, maximum 50 cases, a year, that's the maximum, but usually it's like around 11 cases. Then in 2010, after the 2009 H1N1 pandemic, you have a sharp increase, and then it decreases. People are like, well, why is there suddenly an increase of this basically narcolepsy? What is this narcolepsy type 1, basically? We call it narcolepsy with cataplexy. It's basically what you have is these patients that are having extreme fatigue, and that literally during the day, they're literally just sleeping. They have this excessive daytime sleepiness, and they actually have a lot of basically strong emotions. And what happens is that you have this very, basically this kind of receptors. They're called literally orexin receptors that really regulate a lot our sleep, our melatonin, the way we're sleeping, and also our emotions. That sometimes basically happens is that this orexin receptors are gone. So this is what narcolepsy is. So what they saw is that literally they started to see that, okay, we see that people having 2009 influenza they have this disease. But what they also saw that literally is that these certain kids that were actually vaccinated with the H1N1 influenza A vaccine had not a huge, but had an increased incidence of this narcolepsy. So, for example, these are the number of non-vaccinated and vaccinated ones. If you look at basically narcolepsy cases, you see that there's much more in the vaccinated, basically kind of people. So they're like, okay, we see that people with the H1N1 vaccine, this one specific vaccine, they have a slight increase of narcolepsy, but you also have people that were infected with it also have this narcolepsy. What is it? Well, I have a very good friend of mine, this Tobias Freitag, who published this beautiful paper in Nature Communications. And what he saw was that people that actually, that were basically, that had this NT1, they basically presented, when he took their T cells, they were so reactive towards this neuraminidase and nuclear proteins, so this NA protein and NP protein found in the basically H1N1 basically virus. But this is weird because this NT1 people were not infected with the 2009 and H1N1 virus. They were not. I was like, okay, interestingly wise, this is very interesting. That why are these patients having T cells super reactive towards proteins of the H1N1 virus that they never got infected with? So okay. So basically, he actually took these basically peptides. So these peptides that were basically very reactive in this NT1. These peptides are basically coming from the neuraminidase and also from the nuclear protein. And they saw that basically they actually were very, very similar to proteins that were found in the brain. This basically syntrophin gamma 1 and this pumped, basically this protein O mannose transferase 1. Well, these are found in the brain. So they're like, okay, these viral proteins, NA and MP, are so similar to these proteins found in the brain, the syntrophin gamma 1 and protein and POMT1. It's like, okay, this is very interesting. So what he did, so he literally took the T cells. This is basically coming from NT1. Basically, these are the narcolepsy patients. And here, basically, he gave them this NA, this neuraminidase. This is the influenza protein. And this is basically the POMT1. This is basically the protein found in the brain. So he basically took the T cells and gave them either viral or basically self-protein coming from the brain. And he basically did RNA sequencing on the T cells. And he saw that they got activated the exact same way. It's like, okay, this supports that T cells cross-react, meaning that these T cells are literally, these T cells that are literally activating in the same manner, they're activating in the same manner to both these epitopes. It's like, okay. So what they did is that when they stimulated these two, they did basically TCR sequencing. They sequenced the TCRs, and when they saw that basically the T cells that were really expanding, because we have this clonal selection, T cells that were expanding were basically able to recognize both basically the NA and the POMT1, and this shows like, oh damn, that means that there's a T cell receptor that is able to bind to both POMT1 in the brain and also this NA in the H1N1. This explains why people given this vaccine were having a reactivity, having basically narcolepsy, or the ones being infected. And that basically shows that, hey, this this sometimes can be a bit dangerous. This is not something to show you that vaccines are dangerous. On the contrary, no. People actually that were given the vaccine had a way much lower basically incidence compared to people that were actually infected. It's just to show that how an infection can cause technically basically an autoimmune disease. This is a real case scenario, which they are not coming in the exam. This is just for just to kind of help you understand. So now let's talk about transplantation. So transplantation, as we know, is basically some sort of, let's say, procedure that we use to treat either organ failures, meaning that basically you have a certain organ that's failing, we try to transplant it with a new one, or stem cell transplants used to basically treat immune deficiencies. So if you have certain immune deficiencies, so for example, you have kids that do not have any immune cells working, what you do is you literally transplant a whole immune system. So you actually transplant this hematopoietic stem cells using bone marrow, because the hematopoietic stem cells can create all immune cells. Or if you have leukemia, you remove all your immune cells, because leukemia is basically your immune cells being cancerous. So you remove them, you put basically a new hematopoietic stem cell therapy. Of course, there's a limiting factor that there's a shortage of donors. That you don't have sometimes donors capable for them. So what we see within a transplantation, so if we have basically mouse number A, which has basically the same, basically MHCA, okay? We take a graft and we put it to another mouse that has the same MHCA. These are genetically the same. The graft is tolerated. If we take this basically mouse that has a different MHC, we put it with another mouse that has MHC B, so this is MHC A and B, basically over literally 10 weeks or 10 days, this basically graft, this one is completely rejected. What happens if we do actually, if we take a second skin graft from the same donor to the same recipient, so again, we repeat this from basically 12 days now is basically literally in 6 days. So it's much more fast, more faster. That means that this is mostly T cell mediated or B cell mediated, meaning that there's a memory response. That this mouse now memorized this. So okay, to prove this, what we do is we literally take you take T cells, you transfer you basically now take MH basically this over here, this MHC a mouse, and you take basically a piece of the protein, you put it in a naive MHCB, you do this transplant, and then you also take this guy over here that already has T cells against this, and you inject it, you accelerate this more. So showing you that this T cells really accelerate this kind of rejection. And this is what you see also basically in generally when it comes to real life scenarios. So if you take a piece of your, let's say your skin coming from your legs and you put it basically in your forehead, this is called autograft. So coming from the same individual, you actually have 100% graft. It's actually able to be tolerated. If you have identical twins, this is syngenic, meaning that it's basically different human beings, but genetically the same. So this is called genetically, basically. If you have non-identical individuals, so basically they're not related, this is called allograft because genetically they're not the same. So this is allograft. If you have from different species, for example, from a pig to there, this is xenograft. So the autografts are usually the ones that will be, the graft is tolerated. Nothing's going to happen. It's not going to be rejected. Syngenic also is in generally quite safe. Depends on certain genetic compositions, specifically the MHCs, which we'll be talking about. The allografts, again, you're trying to match people that are genetically very similar. And xenografts, this is extremely difficult, but not impossible. So this is basically will not come in your exam, hence it's in red. I wanted to share this beautiful case because we have an issue, as I said, with transplantations. There's a huge shortage of donors. If you could actually be able to actually make xenografts possible, you would actually can save millions of lives. So what people wanted to do is, and this is coming from University of Maryland. This was in January 2022. I was there actually. I did my my postdoc in University of Maryland, and I was there during basically when they did these operations. I actually was in the seminar. Basically, I forgot the guy. I think this guy's John. His name is the surgeon that conducted this. And what they did was they literally took a pig, and they genetically modified that pig. Basically, they did 10 genetic modifications that basically made the pig's heart look more human and have less basically certain antigens that would induce organ rejection. So they took that pig and they transplanted it. So this pig has now a genetically engineered heart that should be much more human-like, and they basically injected it to this basically to this person that was already having basically a lot of issues. And basically this patient actually survived for around 40 days. They did this again for another, sorry, this patient lived for 2 months. Later, basically, they did this again for another patient, and they basically also survived for 40 days. So what they saw, that, hey, we are almost close to actually creating pigs that we can genetically modify them to have a heart that looks genetically so similar in a way that we can actually create or do transplants. This was a huge breakthrough. And I remember I was in the lecture when they were presenting this, and I was saying, like, we're going places. Like, this is where you see finally human beings doing something good in this world. But things to consider successful in transplantation. So first of all, what makes something actually accepted is two things: HLA compatibility and immunosuppressive drugs. As I said, HLAs, HLAs are the biggest trigger of an autoimmune response because, as I said, HLA molecules are extremely polymorphic. We have so many different types of HLAs. Well, when you're actually, for example, if you have here basically this MHCA and you're transplanting it to here, despite that the organ, let's imagine that you're transplanting, for example, here skin to skin. Well, this skin to this mouse should be the same protein, genetically the same, but and basically the MHC is the same, MHCA and A. Well, let's imagine you have these two mice. This skin of this mouse and this skin of this mouse should be genetically very similar, super similar. But the main thing that is usually not similar is the MHC, because this guy has an MHCB and this has an MHCA. So what occurs technically here is that this mouse, this blue mouse, will create a T cell response against this MHCA. That doesn't have it because that's foreign. That is seen as something as a foreign, basically invader. Well, that's what's happening here with HLA compatibility, because when you take two random human beings and you do transplants, well, the HLA are different, and those are the things that will basically create a T cell response. So an alloimmune response, a different immune response. So you need to make sure that the HLA are compatible. So in the case of here, what you've done is that you're giving it to two basically animals that are literally similar, have similar MHCs. So MHCA to MHCA, great, then it works. If you have different ones, it's not going to work. So you need to make sure that the HLA is perfect. So usually when you do a transplant, when you're trying to find a donor, you're trying to make sure that the donor and the recipient have exactly the same HLAs or very similar ones. Thing is that perfect HLAs do not really exist because it's too polymorphic. It's almost impossible to get it. So what you do is you try to reduce so you try to create you try to find a person that has as similar as possible. So that's what you're trying to do. And then because we do not have perfect HLA match, you need immunosuppressive drugs. These are drugs that are stopping basically T cells from proliferating, T cells from basically activating, anti-inflammatory, stopping inflammations, and basically other biological like antibodies like stopping and antibodies. An example for them is tacrolimus, which is a very famous one. This inhibits interleukin 2. Interleukin 2 is needed as a third signal for T cells. The T cells need interleukin 2 as a third signal to proliferate. So what you do is use actually tacrolimus that inhibits interleukin 2 production. So what happens in the case that you basically have a transplant that is not 100%, that the MHCs are really not 100% there? Well, you have two things that can happen: host versus graft disease, in where the host is actually attacking the graft. So let's imagine me, Firas Hamdan, I get a kidney. So Firas Hamdan's immune cells are attacking the kidney. That's host versus graft disease. Or you can have graft versus host disease, the opposite. This is basically where the graft, so now the kidney is attacking me, Firas Hamdan. So it's the opposite. The host versus graft disease, you see this much more in transplants of organs. The graft versus host disease, you see this more in the hematopoietic stem cells, because here you literally are transplanting a whole immune cells. So this donating basically immune cells that you're giving can attack you sometimes. So we'll basically talk about the two different types. So in the host basically graft disease, this is mostly mediated by T cells. But they can be also basically be done by B cells. We'll talk about this. But the rejection is mostly done by non-self MHC molecules. So in the case basically of, again, the kidney, so the kidney has a different MHC. So when this kidney, you put it in me, Firas Hamdan. Firas Hamdan has a different MHC than the kidney. So what happens is that basically because the kidney has a different MHC, Firas Hamdan's T cells are going to recognize that as foreign, those MHC coming from the kidney. So they will try to kill it. So this rejection can happen through different time scales, and I'll be talking more with that. So this could actually happen literally within minutes to hours. This is called hyperacute rejection. This is literally happening extremely, you do this and you happen within minutes or hours. This is because you have a pre-existing antibody already, basically. This is not T cell mediated. This is T cell mediated. You already have T cells. Sorry, you have basically, for example, me. I already have basically antibodies already present in my immune system, already against the MHC of the kidney I'm going to receive. And then you basically have antibodies that are activating your complement system and they're killing basically the kidney I'm receiving. Or this is acute. This happens days or after weeks, and this is all T cells. What happens is your T cells are now basically taking up your basically the antigens from the MHC. They're presenting it. They're creating an immune response. It's like a vaccine response technically. You're basically teaching your your T cells that hey, this foreign MHC that I just got, you need to kill it. And then there's chronic. This is seen months or years later. This is T cells, but not 100%. It's low grade T cell response. It's mostly that sometimes that the graft is already getting injured. Something is not being perfused with, basically, it's not getting really vascularized. It's getting a bit ischemic, or that the immunosuppressive drugs are really causing a lot of damage to it as well, or causing in generally some damage. So this is much more happens over the years. So basically, when it comes to the acute rejection, what happens is that there's a direct recognition. So what you happens is that there's already basically the donors. This is a direct recognition. So you have the donor APC. So what happens technically is that the kidney has these antigen presenting cells, and they're literally presenting it to my T cells like, hey, this is basically the MHC2 basically peptide. This is the MHC2 peptide. My T cells like, wait, I've never seen this. I need to get activated. Or it's indirectly in a sense where basically my own dendritic cells come in and they literally take basically the donor cell. So this is the kidney cell. They gobble it up. They take the MHC antigens and they present it towards the T cell and to my T cells. So this is indirect, meaning that first you need to kill the donor cell, or no, the donor APCs are presenting it. And then you have graft versus host disease. This is mostly T cells. These are T cells in the transplant that are basically recognizing us, basically the donor, the recipient as foreign. So this is extremely very dangerous, because what's happening is that the T cells, so imagine I get a kidney, and the kidney has T cells that are basically proliferating, and they're going into my body and killing my body because there's the things that they don't recognize. This usually mostly happens when I say in hematopoietic stem cell transplantation. It can happen in graft rejections, but it mostly happens in hematopoietic stem cells, because here you're creating a new whole immune system from a different person, and sometimes they can become a bit haywire against your own proteins. So thank you for listening, and if there's any questions, please let me know.